I don't need help. Okay, here we go. So, back to the march through diversity. Tonight, talking about fungi. They look like plants, they're related to animals, they're very different, they have haploid bodies. We'll talk about all of that. So the lecture, we're going to talk about characteristics and yummy foods and horrible diseases. Actually more of plants than of human and animals, insects than humans. But with global warming, expected to become worse for humans. The major groups that are sometimes defined by fruiting body, do they have sacs, ascomycota, or gilled fruiting bodies, basidiomycota, or sometimes ecologically, do they help plants absorb nutrients? Are they intracellular parasites, or are they symbiotic with lichens? So those are the main groups of fungi. And then, because fungi are sometimes parasitic, this is kind of my excuse to talk a little bit about parasites and the evolution of virulence. Okay, so fungi are part of the opisthecons. That means they, at least some of them, sometimes have a single flagella, and that actually groups them with animals more closely. We share a common ancestor with fungi more recently than either fungi or animals share a common ancestor with like other protist groups or plants. So opisthecons means if there's a flagellum, there's just one, as opposed to heterocons, two flagella. Think like dinoflagellates, which have two flagella, or Chlamydomonas, some of those simple algae, they have two flagella. Okay. The other thing that's kind of interesting is that fungi have cell walls, so you might think plants, but remember, plant and algal cell walls are made of cellulose. Fungal cell walls are made of chitin. We're going to talk about chitin again. It's a polysaccharide found in insect exoskeletons, so another sort of animal-fungus link. Some fungi are our friends. Some of our most important food groups, like wine, and shoyu, soy sauce, a lot of fermented soy products, tempeh, some cheeses, miso, bread, all highly dependent on fungi. So most cheeses are actually bacterial, but especially the blue cheeses, Roquefort, you are dependent on penicillium, a fungus. Some fungi are poisonous. For example, Amanita, the death cap. And we're used to thinking of poisonous mushrooms as being like brightly colored, aposematic, like warning colored red, but not all of them are. So interestingly, Amanita phalloides is not brightly colored. However, it has an odor that might have evolved for a similar reason as warning coloration, to amplify the signal to warn potential, fungivores that this is not something that they want to eat. It's an interesting question whether or not a mushroom should be toxic, and it kind of depends on, it's totally speculative, but it kind of depends, like how tough are their spores? Do they want to get eaten to be dispersed? A lot of the gilled mushrooms, though, are wind dispersed, and so if someone eats their spores and digests them, that's not going to be particularly beneficial. So it's an interesting question whether mushrooms want to get eaten or not. Do they function as fruits, or are they just sort of wind dispersing organs and it's bad for them to be eaten? Anyway, Since we're humans, we have an unnatural bias or interest in human diseases. There are relatively few severe human fungal diseases. Most of the fungal diseases are mild skin infections, athlete's foot, epidermophyton, microsporum, thrush, which in immunosuppressed individuals can be a very serious disease, and fungal pneumonia is a disease that also usually is very serious for people with an already compromised immune system. So part of the reason, and we're going to talk about fungal diseases of plants and insects. In contrast to vertebrates, fungi are often very serious diseases for plants and insects. The story is that the high metabolic rate, high body temperature of mammals and birds is a poor environment for most fungi. So the idea is that high body temperature acts as a fungal filter. But there's this interesting recent idea that as our climate warms and habitats in general might be warmer, that might be creating an environment that is going to select for more heat-tolerant fungi that then might become more serious human diseases. I think that's kind of speculative, but there's an interesting example. A fairly recently described serious fungal disease, Candida auris, is interesting partly because it seems to have arisen in 3 different continents at about the same time. And genotyping has shown that these are independent colonizations of disease-causing Candida auris. Normally, Candida auris is a saprophobe of plants. That means it eats dead and decaying plant material. So the other interesting thing about Candida auris is that many strains of Candida auris are resistant to the 3 most common antifungal medications, which makes it a really challenging, difficult disease to treat. So there's this hypothesis, it's really just a hypothesis, that global warming has created warmer environments in all the continents, and that has selected for more heat-tolerant Candida auris that then gets eaten by birds who are eating the same plant material that the fungi are attacking, and then they poop and they spread it to farms where it can start to colonize people. People move to cities. It's a big problem mostly in hospitals, and where it can spread from patient to caregiver and then to other patients. So just something to keep us awake at night. On the plus side, fungi are involved in several important human medications, pharmaceuticals. Fungi are involved in the processing of more than 10 of the 20 most profitable products used in medicine. Some things kind of make sense because, as we will learn, I might have mentioned it, fungi are, super important decomposers. The other big decomposers are bacteria. That means that there are a lot of habitats where fungi and bacteria are kind of locked in a competitive struggle. And so there are going to be a lot of fungi that produce chemicals that are toxic to bacteria and vice versa. So a lot of our most important antibiotics are fungal in origin. Think penicillin, the first one. Also cephalosporins. But they're also involved in some anti-cholesterol statins and cyclosporin, which is an important immunosuppressant, which is important for organ transplants and stuff. Okay. In contrast to the general trend that fungi are not super serious human diseases, fungi are often severe parasites of insects and plants. And there are multiple examples of semi-recent, the last 150 to 50-year introductions of fungal diseases that have basically wiped out or in the process of wiping out once highly common plants like elm trees, chestnut trees, and certain kinds of oak trees. So I'm going to talk about a couple of those examples. So elms used to be real common tree in North America. Dutch elm disease caused by Ceratocystis ulmi and moved from tree to tree by a beetle. It's wiped out elm trees in North America. This is a dung fly that's infected with a parasitic fungus. We'll talk about a few of those. Chestnut is kind of the classic example of a tree species that has been almost wiped out by an introduced fungal pathogen. Chestnut used to be kind of the dominant canopy tree in the eastern United States. It was incredibly important commercially because it was really fast growing and made, good hardwood timber. It was incredibly important ecologically because chestnuts are delicious and they feed deer and squirrels, all sorts of rodents, and passenger pigeons that are now extinct also. So this is the original range. And interestingly, the roots are alive in large parts of these ranges, but this disease, what happens is a chestnut will germinate, sprout, and the tree will grow for about 15 to 19 years and then die. So chestnut blight as a disease was originally described from New York City trees in 1904. So that means it was probably brought to the U.S. in the late 1800s, the late 19th century, and then introduced in nurseries, and then it spread on its own. So this is a chestnut with a canker developing, and as that develops more, it kills the tree. This is a photograph from 1904 of a chestnut, not 1904, from like the 19-teens, of a grove of mostly chestnut trees after the blight has come through and killed them all. It's really interesting what people have done to try to bring back these trees. And they've used sort of more traditional breeding programs, and more recently have started to use gene editing techniques, genetic transformation. So how does the backcross going to work? I'm not going to hold you responsible for the details. The problem is that, so the interesting thing is, there are trees from Asia. There's a Chinese chestnut that has clearly lived with this fungus for millions of years probably, and the fungus doesn't kill Chinese chestnut. Chinese chestnut can tolerate this blight. Unfortunately, the Chinese chestnut is a little different. It doesn't grow as quickly. It's not as good like a carpentry hardwood. And so we've planted some Chinese chestnut here, and those might well spread, but they actually are not as competitive trees in the U.S. So like the American chestnut does really well on its own, grows fast, is a good competitor in the forest. The Chinese chestnut is not going to fill that niche in the same way. So there have been a few programs, first starting in 1921, then again in 1986. The idea was you cross Chinese chestnut with American chestnut. Now you have offspring that are hybrid. And the idea is you then take some of those hybrid offspring and you cross them to each other. And some of them should have both resistance genes from the original Chinese parent, but more of the other traits from the native parent. And the idea is that you do that, and then you back cross to the American, then to create a number of hybrids, then you dihybrid cross again, to get at each stage, and then you do selection with the chestnut blight, pick the ones that are most resistant, and you keep doing that and you get resistance against a background of your original species. The problem is that resistance to this blight in the Chinese chestnut is polygenic. It takes at least 3 different loci having the right resistance genes. So you need a huge sample size to get some of those dihybrid crosses to have both copies at all 3 loci. And the other problem is that it usually takes 19 years to tell whether your tree is resistant to blight or not. Like, you grow it and then you see if it lives or dies in 19 years. So for a number of reasons, that crossing program didn't succeed. A more recent program that is also going to take a while but might have a little bit higher level of success is a different approach. It's known that oxalic oxidase, a gene that digests oxalates, can defend some plants against certain fungal diseases. So an oxalic oxidase gene that conferred fungal resistance in wheat, to a different pathogen, but in wheat, was taken, put into plasmids and used to transform the genome of, just inserted into American chestnut genome. And then those plants are tested. And the other part of this more modern program are assays for this oxalic oxidase trait that can be done on young plants, just a few week old seedlings. And so that the combination of being able to assay quicker and also being able to just insert the gene into the tree with the traits that you like, hopefully will yield better results more quickly. What is this graph showing? This graph is showing, on the x-axis we have weeks after inoculation with the chestnut blight. Then we have 3 lines. Chinese chestnut are the diamonds. And on the y-axis, we have the area of canker, the size of the blight infection. If it's bigger, that's worse. The Chinese chestnut and this transformed chestnut, it's called Darling 4, with wheat oxalic oxidase gene inserted. Both of those, even after 14 weeks, have a very small amount of fungal canker, which usually means they're going to live for quite a few years at least. Whereas the American chestnut, large canker developing after, 3 months. So this is probably, a more useful approach. Okay.
I have a question.
Yeah.
Can you just clarify the relationship of the Chinese chestnut with the American and Japanese?
So American and Chinese chestnut, 2 different species. They're related. They both make, nuts that you can eat. They're a little different. They happen to have some traits that differ, particularly like speed of growth and competitiveness in the wild, that make the Chinese chestnut not ideal to just plant in the U.S. to replace American chestnut. But the Chinese chestnut has lived with the blight for probably millions of years. It's resistant. So, perhaps both. The best guess from one paper was that blight was introduced by chestnuts that were imported from Japan in 1904, but Chinese chestnuts are known to be resistant to chestnut blight, and chestnut blight is found in both widespread in Asia and in Japan. Yeah. Yeah.
You said there were different species, right?
Yeah.
The Chinese chestnut and American. How are they planning on doing the dihybrid cross?
Oh, yeah. They're different species. Aren't species not able to form fertile hybrids? And the answer is, yeah, species concepts and species definitions are bad. They're considered separate species, but yes, they can be hybridized, at least in a captive breeding situation. In other words, I actually don't know enough about chestnuts. That's a great question, and I should go look up. whether they hybridize in the wild. But plant breeders can hybridize plants that aren't able to hybridize in the wild, because sometimes the isolating mechanism is something prezygotic, like they flower at different times of the year, or they attract different insects. And if you're a breeder, you can easily overcome that, right? But I don't actually know in detail if this is the case. So they're thought of as different species. They've got a whole bunch of different traits. They might be able to hybridize. That could be because people are sloppy about defining separate species. It could be because they are good species, but you can overcome it. Fascinating fungal evolution fun fact. It was discovered after visiting Belize that one of his CDs was discolored and unreadable, and it turns out that the aluminum and polycarbonate layers of the CD were attacked by fungal hyphae. So now there are some fungi that can gain energy and live on plastics, polycarbonates, some new molecules that were created by humans. That's also bacteria. Some bacteria have evolved that can break down new polymers in plastics. Fungi have some of those tricks as well. Okay, so I already said why fungi are the most important good thing about fungi, their most important ecological role, but let's see it.
These organisms, which thrive on decaying matter, fill a vital role as the major decomposers in our ecosystem.
So this is a time-lapse photo, video that's taken by taking pictures every few minutes over several days of fungi breaking down an apple or a loaf of bread. There's kind of a trick to these time-lapse. You have to do it in a very humid room. You stick these in your bowl at home, they might get moldy and a little disgusting, but they won't get this totally awesome. But if you crank up the humidity, you can get your bowl of fruit to look like this. Anyway, fungi are incredibly important because without decomposers, the nutrients that are trapped in the bodies of dead organisms won't become available to plants and animals, and nutrient cycling is going to be much more stulted. And actually we'll get things like fossil fuel deposits, right? Why do we have fossil fuel deposits? Because hundreds of millions of years ago, the fungal decomposer community was not as well developed as it is now. Like, we're not going to make more oil in the next 100 million years because when our plants die, they get broken down by fungi more rapidly and recycled into new life. It's kind of interesting. Anyway.
In this sequence.
Whoa. Awesome. We don't have to see it again. We can find fossil records of fungi if we are careful to know how to look. So sometimes we can find preserved spores in amber. Sometimes we find actual, mineralized fossils that are only consistent with certain kinds of spores or certain mycelia. The body of a fungus is called a mycelium. A mycelium is like a group of hyphae, of tubes. I'm using these words because we want to have all these words by the end of the lecture, but it's in a slide, like 3 slides from now. So this is kind of cool. What is this figure showing? It's showing that, first, fungi and animals split about 900 million years ago. Remember, 543, that's the Cambrian explosion. But land plants started only a little bit before that, 600 million years ago. The oldest fossil fungi, for molecular clocks, the Ascomycota and Basidiomycota split 600 million years ago. And there are fossil records of glomales, which are the mycorrhizal fungi, the fungi that help plants absorb nutrients from the soil. They're pretty much the same age as land plants. And this that's what this Fungal from the Start, that was the title of this science paper, is saying, that the fossil record and molecular clocks indicate that mycorrhizal fungi are just as old as land plants. And it's possible that fungi were needed for plants to originally colonize land, which kind of makes sense because the earliest land plants don't even have roots, right? They don't have true roots. That comes with vascular plants. The bryophytes don't have true roots. Hornworts, liverworts, stoneworts. So fungi were probably really important in absorbing nutrients. Okay, so fungal characteristics. They have cell walls. They almost never have flagellae. We'll talk about the chytrids. They have flagellae. The cell walls, however, are made of chitin. Totally different than plant cell walls, showing their closer affinity to plants. Fungi are mostly multicellular. Unicellular fungi are called yeasts. Fungi are chemoheterotrophs, like us. However, they are primarily decomposers, often parasites, occasionally predators, which is kind of interesting. We'll show a few predatory ones. And they're often mutualists, especially with plants. So, fungi usually have external digestion. Their body structure are these tubes, these hyphae. They secrete digestive enzymes that break things down, and then the hyphae absorb nutrients. The hyphae are those specialized structures for absorption. Hyphae come in sort of two flavors. They're either divided up into sort of individual cells. Those are septate hyphae. We're learning these words. Septa means, division. Or they're coincidic, a single tube with kind of free-floating nuclei moving within the hyphae. An example of a predaceous fungus is Arthrobotrys. It's a soil fungus. And it has hyphae that are specialized to capture and kill nematodes. So they've got these little 3-bead rings that if a nematode crawls between, they'll sense, they'll swell up and trap it. And it's a really interesting thing. Now that we can use stable isotopes, I don't think I have a slide, but now that we can use stable isotopes to identify like sources of different molecules like proteins, like nitrogen, it's been determined that like some trees get something like 60% of their nitrogen from springtails. Springtails, what are springtails? We'll talk about them in the arthropods. They're a primitive flightless hexapod, collembolans. The springtails are trapped by fungi that absorb the nitrogen from the springtails or from nematodes, and they transfer that nitrogen to their symbiotic, the trees that they live with. The trees use that nitrogen to make more cells and then photosynthesize and give the fungi sugars and energy. So it's sort of an exchange that way. This is just a closer-up picture of the little trapped, swollen nooses in Arthrobotrys. These are fungal hyphae attacking a bacterial colony. So I've tried to use a lot of these words, so make sure that we feel good about them. We'll, the next couple slides, define them, but a spore is, yeah? Sorry, what? It's a good question, whether you think of bacteria as like, you think of, it's nematodes definitely predation. You think of predation as eating animals or whole organisms. So it could be predaceous. Yeah, it is a kind of predation, totally. Hyphae are the tubular structures of that fungi are composed of. A mycelium is the body of the fungus. It's basically the collection of hyphae that make up the body of the fungus. A haustorium is a part of a bacteria that penetrates cells. And sometimes this can be parasitic, this can often be symbiotic. So in the glomales, the mycorrhizal fungi that live with plants, the haustorium is sort of the structure that enters the plant, either between plant cells or into plant cells. Yeah. It's part of the fungus, yeah. It's kind of the penetrating part of a fungus. Hyphae, like I said, might be undivided. Those are coincidic. They might be septate, divided by septa. Understand, in general, when we're thinking about body types and life cycles, we're going to go over this a bunch. Animals were diploid. Plants alternate generations. They have diploid diploid sporophytes, haploid gametophytes. Fungi, by and large, are haploid. They have haploid bodies. However, fungi will have this interesting state where if two complementary mating types meet each other and love each other very much, they can fuse, but the nuclei don't fuse. So when the cells fuse, that's called plasmogamy. However, they will then enter a state called the dikaryotic state. That's when each cell has two haploid nuclei. And they'll do that. They can sometimes grow for a long time in this dikaryotic state. And then, usually when times get rough, then they will make a fruiting body. They'll do meiosis. The nuclei will fuse. They'll produce haploid spores that are different than either of the parent nuclei. And those will go on to produce more haploid fungal bodies. Okay, so like I said, mycelium is a network of hyphae. The mushroom is actually always just a small fraction of the fungus, right? The mushroom is the fruiting body of some kinds of fungi, Aschimycota, and especially gill-forming Basidiomycota. They make mushrooms. But that's not the body. The mycelium is actually this network of tubes that's actually like in the soil or eating out the inside of the tree. Interestingly, the world's largest organism, maybe one of its oldest too, is a fungus. Amarillaria ostoyae was identified because there was a grove of diseased trees. It infects ponderosa pine. Happily, we use pine to make tongue depressors and ice cream sticks. So this kind of population structure is studied by sticking tongue depressors into the soil over several square miles. They get attacked by fungi, and then you genotype them and see. And what was discovered when the DNA was analyzed was that a single organism extended clonally, genetically identically, over 2,200 acres, 3.4 miles in diameter. The estimate by doing careful extractions in a few square meters and then multiplying out was that the fungus probably weighed about 100 tons. And based on sort of reasonable estimates of growth rates, it was estimated to have been 2,400 years old if it had grown out fairly uniformly from a single spore. So that's kind of cool. Fungal life cycle, like many organisms, even some animals, lots of plants, asexual. In good times, you're just doing mitosis. You might make haploid spores. However, often in response to stress, there will be sexual reproduction. So the nuclei will fuse, you'll get meiosis. That produces zoospores. So it doesn't produce gametes, it produces zoospores that then produce new haploid offspring, mycelia, and they might then fuse with different mating types, and so sexual reproduction happens that way. So you don't have gametes making zygotes and diploid bodies. No, you don't have that. You just have haploid bodies. And this is what I said. Anyway, most of the time you have asexual reproduction. You might have spores forming asexually. Those will produce haploid mycelia. When two mycelia of different mating types, that's why you probably read like, there are fungi that have 8 different sexes. And you're like, 8 sexes, what? That means there are 8 genetic strains that won't do plasmogamy, won't fuse with their own strain, but will fuse with any of the 7 other lineages, the 7 other strains. So they're not different sexes in the way you might be thinking of different sexes. They're just 8 complementary mating types, and all of them won't mate with themselves, but they will mate with any of the other 3 or 5 or 7 types in that species. So when complementary, that means different mating types meet, the cells will fuse, and then at some point the nuclei, and then they'll have this dikaryotic stage with 2 separate haploid nuclei. Then at some point the nuclei themselves will fuse, making a zygote that doesn't develop into a body. In fact, it immediately undergoes meiosis to make haploid spores. The difference between asexual and sexual isn't that one makes gametes and the other doesn't, is that the spores from meiosis are different than either the parental types. The spores in the asexual phase are identical to the single haploid parental type. Okay, so what I just said, fusion of cells is plasmogamy. Fusion of nuclei is karyogamy. Remember, eukaryote means true nucleus, so karyon is nucleus. Karyogamy is fusion of nuclei, going from 2 haploid to a single diploid nucleus. Once you have a diploid nucleus, now you can have meiosis and make spores that are going to be meiotic spores that are going to be different than either of the parental types. Okay, so now we're ready to talk about the different fungal groups. Not going to talk about them much, but microsporidia are within cellular parasites of animals. They're really simplified cells. They're among the smallest eukaryotes known, and they're kind of interesting. Chytrids are the true water fungi, the true fungi, water molds, in contrast to the oomycota that we learned about before. The potato blight, that was caused by an oomycete that looks like a fungus but has cellulose cell walls. Chytrids are water molds. Interestingly, both they make zoospores and gametes that have flagella, so that's only found in the water molds. We're not going to see flagella or gametes in anything else. Glomeromycota are the arbuscular mycorrhizal fungi. So these are the, also sometimes called endomycorrhizal fungi. These are the fungi that penetrate the cells in plant roots and aid in nutrient absorption. So that's kind of cool. So microsporidia, chytrids, glomeromycota, we've defined those mostly ecologically. You know, intracellular parasites, water molds, root mycorrhizae, symbionts. The zygomycota, Aschimycota, and Basidiomycota are defined by their fruiting structures, that they form zygospores in the zygomycota, sacs with ultimately 8 spores each in the Aschimycota, and gilled fruiting structures in the Basidiomycota. So this is a phylogeny. Chytrids and zygomycota are a little bit hard to place. Microsporidia, kind of the more different than anything else, these tiny parasites. And then we have the zygomycota. And then the 2 groups that have a dikaryotic stage, 2 haploid nuclei in each cell. That's a separate stage from fusion to make to have diploid cells that'll then undergo meiosis. So the ascomycota and the basidiomycota are most closely related to each other. They're the only ones with that 2 haploid nucleus stage. And hyphae that separate into separate cells, septate hyphae. So we'll talk a little bit about each of these groups. So the water mold's unique in that they have flagellated gametes. And so they used to be like their own protist group separate from the fungi because of the flagellated gametes, but now they're lumped in with the rest of the true fungi. So you can see haploid zoospores and the male and female gametes that fuse. These are both have flagella. The other thing that's different about chytrids than other fungi is that they can have a diploid body. So I said before, plants are the ones that have alternation of generations. Chytrids, the weirdest fungi in terms of life cycle, they're the only ones that have a diploid body. So odds are on an exam, I'm not going to show you chytrids because they're weird. I'm going to show you Aschimycota, Basidiomycota, Zygomycota. Those are the ones that are much more typical of fungi that only have haploid bodies. And it's really kind of tricky comparing chytrids, which are true fungi, and the Oomycota that include potato blight that don't. So there are differences in that oomycotes are diploid, whereas real fungi are haploid or dikaryotic. Oomycota, like potato blight, have cellulose cell walls, whereas chytrids and other fungi have chitin cell walls. And there's a difference in the structure of the cristae and the mitochondria. Okay, chytrids are actually really a subject of intense research because they are most likely responsible for many recent extinctions and declines in amphibians worldwide. And it's been an interesting story, and it's one that's really changed. Since I was teaching this 5 years ago to now. So these are the older slides, and then I'm going to show you some newer stuff. So one thing, amphibians in general are kind of ecological canary in the coal mine because they live in 2 habitats, right? So if there's something in the water or some terrestrial insult, either of which can harm amphibians. So in some ways they're especially vulnerable. And some things we know are bad for amphibians, like loss of habitat, pesticide toxins, increased UV exposure. However, there have been several recently documented declines or extinctions of amphibians at high altitudes in undisturbed forest that weren't being subject to like pesticides or other pollutants that we knew about. Example being like the golden toad in Costa Rica. So parasites seem to be especially important for some species, and one species that's been identified is Batrachochytrium dendrobatitis, or BD. And this is a figure from a paper this year in Science showing the numbers of species of amphibians that have gone extinct due to associated with infections of chytridiomycosis. So one thing that's interesting is a lot of extinctions, a lot of affected species in Central America, South America, less in North America, less in Europe, and almost none in Asia, less in Africa, And that's kind of interesting. We're going to come back to that. So there was first, a lot of research was needed to actually show that this chytrid parasitic fungus might actually be causing these declines. The clue that it was probably parasitic and not some of the other possible causes is that some amphibian populations were crashing rapidly in undisturbed habitats, so those populations weren't going because of habitat loss or because of pollution. So this chytrid was identified recently, 1998. That's really recent. Feels less recent to you. Feels like yesterday to me. Koch's postulates, as you might know, are that take 4 stages. You have to first isolate the possible parasite from sick individuals, and all sick individuals have to have that parasite. You then have to be able to grow that parasite in culture, use it to reinfect healthy animals and cause that disease again. And then finally isolate it from those new sick individuals. So that's Koch's postulates, and those were applied to amphibians and showed that yes, BD can kill amphibians. Doesn't explain where it came from. So there was one hypothesis, that's called the out of Africa hypothesis, and it centers around this species called Xenopus laevis, the Xenopus frog. If any of you go on to study developmental biology, especially in Professor Weinstein's lab here, but all over, Xenopus is like a model species for studying development. But before it was used to study the early cell patterning and cell communication that determines cell fate early in development, Xenopus was, which is a South African species of frog, was grown and shipped all over the world as a pregnancy test. So the cool thing about Xenopus is that if you take urine, you can take a urine sample from a woman, and if she is pregnant, that urine will induce spawning in the Xenopus frog. So pharmacists would keep a bunch of Xenopus frogs in the back and take one out, put it in a tank, and that was one kind of early pregnancy test that people used in the early 1900s. Xenopus is not killed by BD, and interestingly, Batrachochytrium dendrobatitis was identified from a museum specimen of Xenopus from 1938. So this is a 2004 paper, and it was arguing, BD was spread out of Africa from by frogs that were being shipped all over to be used in pregnancy tests. Interestingly, it's a great theory, supported by some initial, suggestive evidence, and then it was tested and it failed to be supported by other lines of evidence. So in the textbook, there's a little box that describes an experiment, a survey that was done to ask whether the disease-causing forms of BD found in North America are closely related to the strains of BD that are found in Africa. And the answer is no. So pathogenic strains of disease, the red ones are disease-causing BD isolated from Western North American amphibians. And sometimes they appear to have come from Africa, and other times they're closely related to other American strains. So, no, there doesn't seem to be evidence from this survey, an example that's described in the textbook, that disease-causing BD came from Africa. And since then, there's been a really good study, this is from 2018, showing that disease-causing forms of Batrachochytrium dendrobatitis are by far most genetically diverse in Asia. What does that tell you? Well, if you remember what we learned about molecular clocks, the idea that most substitutions, most changes in amino acid, in base pair substitutions are neutral, and so you can use them as a clock. And that means if a population has been in an area for a long time, it will have accumulated more genetic variation, more base pair substitutions. So when you're looking at a bunch of populations, and some are all pretty genetically uniform, that suggests that that population has been in that geographic area for a shorter amount of time. It more recently descended from a common ancestor. And that's what was shown for Batrachochytrium dendrobatidum. It seems to have originated, the disease-causing form seems to have originated in Asia. They were spread around, over the last at least couple centuries, and now are causing amphibian declines, sort of worldwide. Okay, so that's it for water molds. Zygomycota. Zygomycota includes common molds, things that, the stuff that ate up the bread and apples in the movie. They have coencytic hyphae, so that means the hyphae are not divided into cells with septa. The sporangium, the spore-forming structure, develops through meiosis and that makes haploid gametes. So basically, the heterokaryotic stage is short. You don't have like a long dikaryotic, heterokaryotic stage. What you have is complementary mating types form. They make this zygospore that undergoes meiosis, and make the zygosporangium that undergoes meiosis and then makes spores. Some important, so you can see common molds, stuff that eat your strawberries and your bread. Many insect diseases. This is a flower fly killed by a zygomycete, although there are other fungal groups that cause insect diseases. And they have this short phase. Plasmogamy leads to a zygosporangium that then leads to karyogamy, fusion of nuclei, making zygospores that are haploid but different because they were caused by meiosis, and then you get coencytic haploid bodies forming. So that's the life cycle of Zygomycota. And this is the same thing, but this might be easier to read. There's some really weird, interesting zygospores. So this is a species that grows on dung. Dung, it's like it's one plop of poop, and then they've got to get to another one, or maybe to a cow, or maybe to a grass leaf that then will get eaten by a cow. That's its life cycle, and then it can get, the spores can, grow, then develop within a new dung ball. But they have to get off that dung patty, because that dung patty is a dead end. And they have a subsporangial swelling that focuses light, heats up, and causes them to fling their spores long distances. Distance of a few meters, which for a spore is pretty far. And I think I have a video of this.
Cows don't eat their own muck. How do the spores of these fungi get into the cow in the first place? Well, this one has its own incredible little device. The black spores at its tip are sat on top of a miniature water cannon that fires the spores beyond the ring of grass that has grown up around the cow path and that the cow won't create.
Yeah, the cows don't like eating poopy grass, so that's the challenge. They have to get their spores onto clean grass.
The spores that bombard the pasture are very sticky. It will only be dislodged when the cow eats and digests the palatable grass. But there are organisms that want to get inside the cow. Parasitic eelworms need the cow's warm insides to complete their life cycle.
So these are nematodes.
And to hitch a ride on a jet-propelled fungal artillery shell.
They climb onto the sporangium, and then they get flung as well. Cool. Okay, so that's it for zygomycota, bread molds, and some interesting diseases of insects and plants. The glomeromycota, or the arbuscular mycorrhizae, are incredibly important for aiding nutrient absorption in plants. So endomycorrhizae penetrate the cells of plants, and then they also reach out into the soil. And they're really important for absorbing lots of things, water, but especially phosphorus. So in phosphorus deficient soils, these fungi are going to be incredibly important. There is also another group of fungi that aid in nutrient absorption, and those are called ectomycorrhizae. Those penetrate the roots, but their haustoria only travel between plant cells. In contrast, arbuscular mycorrhizae, their haustoria penetrate into the plant cells. They can both be beneficial for plants. They just, originated independently, and they have a different way of growing in association with plants. But what you can see is if you don't allow the plants to have any fungi, you use antifungals, or you grow them in sterilized soil, soil that you stick in the microwave for 20 minutes, and then you have it sheltered so that no more spores can get in, plants will grow more slowly. So this has been shown over and over again in lots of different experiments. These are pine seedlings with mycorrhizae, without mycorrhizae. Mycorrhizae are important. Here you can see a cross-section of a root with the mycorrhizae growing on the outside. Here you can see the feathering mycorrhizae growing out from a pine seedling's roots. Here's another experiment showing that especially in low phosphorus soil, plants with mycorrhizae are growing much more robustly. Okay, yeah?
Sorry, this is about something like 3 slides ago. Can you, again, distinguish arbuscular and ectomycorrhizae?
So, the true glomeromycota are just the arbuscular, also sometimes called endomycorrhizae. And structurally, the haustoria, the penetrating part of the fungi in arbuscular or endomycorrhizae, actually enter individual plant cells. Ectomycorrhizae stay outside of the cells. The haustoria penetrate the root, but they grow in between the plant cells and don't actually enter individual plant cells. Okay, ascomycota or sac fungi, diverse group, including some yummy mushrooms like the common morel, important yeasts that help make bread and beer, and some other fungi, cup fungi. So now we're looking at the two groups, the Aschimycota and the Basidiomycota, that in some ways are pretty similar to each other. These are the two the two septate groups. These are the two fungal groups that have septate dividing the hyphae into individual cells. So you have haploid bodies, and when two complementary mating types, remember there can be up to 8, I think, love each other very much, they might fuse their cells. That's plasmogamy. That's not the same thing as fusing the nuclei. So you can have a dikaryotic mycelium, a body of these not diploid but 2 haploid nuclei cells, and then at some point when they're getting down to forming a fruiting body, they will undergo karyogamy. They'll make in Aschimycota, they'll make these sacs. In Basidiomycota, these will develop at the end of gills. They'll make these sacs. The nuclei fuse. They undergo meiosis. That will then produce 8 different haploid ascospores per sac, and then those go out and make new haploid bodies. And there are some important medicinal as well as disease-causing fungi, including cyclosporin, which is an immunosuppressant drug, and one little vocabulary thing, conidia are the asexual spores of Aschimycota. Kind of some trivia. Conidia of, I'll talk about it later, of one Aschimycote, Bovaria bassiana, are a really valuable, natural, biologically based insecticide because they attack several pest insect species. A severe disease in the past called St. Anthony's fire or ergotism, it would lead to especially like fevers and hallucination, is caused by an Ascomycote ergot Clavipes purpurae, whose fruiting structure, these ergots, replace some of the seeds in grains like barley and rye. So this is rye that's been infected by ergot, and if you make flour out of this fungally infected rye, you can get some nasty disease called ergotism, accompanied by gangrene, nervous spasms, psychotic delusions, and convulsions. So people with ergotism were sometimes accused of witchcraft or, listened to like prophets. There are a number of entomopathic Aschimycota. So the conidia, those asexual spores of Bovaria bassiana, can be incorporated into biologically based insecticides, which are nice because they're not really toxic to us. This is actually the species that I study, Colorado potato beetle, turned into cotton candy by Bovaria bassiana, which penetrates the cuticle. Actually might be useful against both like bed bugs and malarial mosquitoes. It's an interesting fungus. Basidiomycota are the gill forming fungi, and this is the majority of like the edible mushrooms. Most of them, and the highly toxic ones like Amanita, are Basidiomycotes. This is a giant puffball. This is the only mushroom that I consider myself qualified to identify because it's really easy to recognize. It's like the size of a soccer ball or a basketball. It looks like a giant brain. It is edible and does not taste that good. So I've harvested them in the wild, cooked them up, and been the only one in my family to eat them, and there you go. So Basidiomycotes, similarly haploid, septate bodies, really very similar to Aschimycota, except what's different is they make these gill structures that then lead to the basidia on the ends of the gills. Karyogamy, fusion of nuclei, leads to meiosis, and then new haploid spores. And these are the gill-forming fungi. If I'm a good person, no, I'm not a good person. I forgot. It's going to bring in mushrooms so we could do, extract the spores from gill-forming mushrooms. The sort of print that you can make is characteristic of each species. Makes its own sort of spore print. Basidiomycota also include the puffballs. These have wind-dispersed spores, which are kind of cool. There's some Basidiomycota, including the shelf fungi. So you see the fruiting structures growing out of the trees, but in the soil, some of them will be attacking nematodes. There are also insect parasitic fungi within the Basidiomycota. So several groups have insect diseases, although the Aschimycota are the ones that make this useful insecticide. Other Basidiomycotes include horrible stinky ones that attract flies to distribute the spores, and bird nest fungus that's used in bird nest soup, with specialized dispersal agents that get basidiospores that get splashed out and scattered with raindrops. The last fungi I want to talk about are lichens, which are symbiosis between algae, usually for photosynthesis, and fungi for structure, anchorage, support, and shelter. So in some places, lichens are kind of like the dominant plant form in the high Arctic. Lichens can often grow in habitats that are too harsh, too cold, or too dry for regular plants. So you see them growing on the surface of rock that can get incredibly dry, and in the Arctic where it's incredibly cold. Lichens often reproduce asexually through structures called soredia, which are basically bundles that contain both fungus and algal cells that can break off and then form a new colony. I think I'm going to end there because the last segment here is about mutualism, parasitism, and the evolution of virulence and disease. And I kind of want to have a fresh 15 minutes to develop that. So we'll talk about that next Thursday. And we will try to get the exam back to you in lab next week. It may or may not happen. It might take 2 weeks.
Uploading the essay.
Yeah.
Where do I do that on Blackboard?
That should be in your lab section, but I noticed today that even though I sent out instructions, no one did it.
Yeah.
So I need to get them. I got